My name is Diedre Reitz, and I'm a postdoctoral fellow at the University of California, Davis, mentored by Drs. Wolf Diedrich Heyer and John McPherson. I study a group of proteins called the mismatch detection proteins and their role in preventing cancer. We often think of our DNA as a kind of genetic language, with each DNA base representing a single letter and the genes or regulatory elements representing words. In this way, you can imagine your DNA as a book. This book tells your cells how to make proteins, which you can think of as the building blocks of life. But what happens when this DNA is damaged? What if the words contain spelling errors or typos? What if a sentence is moved from the very end of the book to the beginning or the middle? The DNA in our cells is constantly incurring damage, which can lead to errors like these. Sometimes this damage comes from external factors. You probably know that you need to wear sunblock on a sunny day. That's because the UV from the sun can cause huge amounts of DNA damage. Over time, this DNA damage can lead to cancer. In addition, some normal cellular processes can also introduce errors into our DNA. For instance, errors can occur during cell division, when the cell will copy its DNA in order to pass this DNA on to a new cell. Given that the cells in our body are constantly experiencing DNA damage, why don't they all become cancerous? Why is it that some individuals can survive nearly their entire lives without being diagnosed with cancer, whereas others will be diagnosed with cancer at an early age, or sometimes experience it more than once? Well, our bodies have evolved multiple pathways to repair or tolerate DNA damage, preventing us from developing cancer every time there's an error. So if our bodies have the ability to repair this DNA damage, why do we get cancer at all? One common theme is that the proteins responsible for repairing this DNA damage become altered. These alterations cause the DNA repair proteins to become non-functional. Since the cell is no longer able to repair this DNA damage, errors accumulate. My research is focused on a family of proteins called the mismatch detection proteins. The mismatch detection proteins scan the DNA in our bodies, looking for errors like these. They are involved in two major DNA repair pathways. First, the mismatch detection proteins find a kind of error in our DNA that we can think of as a spelling error or a typo. Our DNA is made up of 4 repeated bases: A, T, C, and G. Usually, A pairs with T and C pairs with G. However, sometimes an A base will pair with a C or a G and not a T. Alternatively, a DNA base can become erroneously inserted into the text or deleted from it. The mismatch detection proteins find these errors and work with other proteins to correct them. The second DNA repair pathway in which the mismatch detection proteins are involved is called recombination. Recombination can fix several different types of DNA damage or errors, such as when there are large gaps in the genetic text where many words are missing. Recombination uses an unbroken region of DNA that perfectly matches the broken one to repair the DNA damage in a copy-paste type reaction. Normally, there are 2 perfect copies of any given gene. If one copy is damaged, the other copy is used as a template to guide the repair process. But the DNA repair proteins aren't perfect. Sometimes, instead of selecting a region of DNA that perfectly matches the broken one, the DNA repair proteins select a region of DNA that's similar to the broken region, but that isn't an exact copy. When the broken strand pairs with this template, since the two do not perfectly match, this creates a kind of error in the DNA, which the mismatch detection proteins will discover. Once they find these errors made by the DNA repair proteins, the mismatch detection proteins reverse them. This is critical because if these errors made by the DNA repair proteins were not corrected, it could lead to a rearrangement. Rearrangements happen when a sequence of genetic words, maybe a sentence or even a paragraph, is moved from the place where it belongs to some other place. Similar to rearranging sentences in a book, this error changes the meaning of the genetic text. The mismatch detection proteins are involved in two key DNA repair pathways. In both cases, these proteins act as proofreaders, finding errors in our cells' DNA and correcting them. Without functional mismatch detection proteins, DNA errors accumulate and cause cancer. In fact, a significant fraction of cancers affecting the skin, colon, bladder, and endometrium have alterations to these proteins. Most cancer patients with alterations to the mismatch detection proteins acquire these alterations randomly and in one specific area of their body, which gives rise to the tumor. However, some individuals have inherited alterations to these proteins. In these patients, there isn't a complete set of functional mismatch detection proteins, so errors accumulate. This condition is known as Lynch syndrome. Patients with Lynch syndrome often experience cancer multiple times during their lives, with their first diagnosis coming at an early age, sometimes as young as 2 or 3 years old. Better understanding the roles of the mismatch detection proteins in these two DNA repair pathways will undoubtedly improve the quality of care that we can give to these cancer patients. I'm interested in understanding which of these DNA repair pathways is affected in cancer patients with alterations to the mismatch detection proteins, and whether loss of function of one DNA repair pathway or the other correlates with the aggressiveness of the tumor or its location in the patient's body. To gain unprecedented insight into the functions of the mismatch detection proteins, I am working with a team of investigators at UC Davis. We're using a combination of innovative DNA sequencing technologies, genetics, and biochemistry to address these questions. I hope that our groundbreaking approach will help us understand what the best treatment options are for cancer patients with alterations to the mismatch detection proteins, and that our work will lead to improved quality of life for these individuals.